All right, we don't want to take time away from the speakers, so if everyone could get seated. So my name is Ben Stanger. I'm a gastroenterologist at the University of Pennsylvania. I'm also on the Scientific and Medical Advisory Board for the foundation. It's my pleasure to moderate, along with Daniela, this session, which is dedicated to genomics and epigenetics of cholangiocarcinoma. So I'm just going to get right on with it and introduce our first speaker, who's Maia, who is currently at the Mayo Clinic. His focus is on functional epigenomics and how enhancer regulation has implications on cellular heterogeneity and treatment of malignancy. He's won numerous awards. I won't go through them all, but really very, very accomplished scientist, and we're looking forward to hearing his talk, which is about epigenomic changes in cancer development and progression at single cell resolution. So, pleasure out.
Thank you. Thank you for the invitation. I'm delighted to be here. I got the opportunity to come to the dinner yesterday and meet some of the patients. This is really an inspiring meeting. Full disclosure, I don't work in cholangiocarcinoma, but I hope that with the techniques and the analysis that we are doing in the lab, you can get inspired and potentially start collaborations. So these are my financial disclosures. So in the lab, we are very interested in studying the epigenome. I hope I don't have to convince you that the epigenome is very important in cancer. But even though in my previous research as a student and as a postdoc, I was focused mostly in histone marks and histone variants that pretty much shape the way the epigenome can be read. We now in the lab are also very interested in associating those histone marks and histone variants to other patterns such as DNA methylation, which is illustrated there. And one specific pattern that we're really interested in the lab is to try to understand the transcriptional regulation through looking at enhancers. So enhancers can be also called regulatory elements. These regulatory elements are present in the genome and are quite important. And what is one important feature in enhancers that is important to note is that these enhancers are basically binding sites for transcription factors. Transcription factors will recognize DNA-specific sequences and therefore bind to these regions and act towards activation and repression, depending on a lot of different features such as cofactors and also their own proteic domains. And so one thing that really makes this analysis really challenging is that enhancers, they are located throughout the genome, as I said, but they actually can have impacts in gene expression of genes that can either be nearby in cis, or they can actually be very distant. And so when we are looking at features and we map these enhancers in a very cell-specific manner, we always struggle to try to understand whether those enhancers are affecting gene X or gene Y. And that is really one of the things that by mapping the epigenome at a large scale using a variety of different methods, we are trying to really accomplish a much more holistic perspective on how these things are regulated. So as I said, these regulatory elements are quite abundant. In the human, we can now define them close to 8% of our genome, which if you compare to the loci that are associated with genes, it's just 1%. And so this variety of regulatory elements that are spread in the genome can be really traced back not just by the sequence itself, but also by these patterns that I mentioned, that they are basically the basis of the epigenomic analysis. And those patterns are histone modifications or histone variants. The histone modifications, probably some of the ones that you know, is H3K4 trimethylation, which marks promoters, H3K4 monomethylation, which typically marks enhancers, and so on and so forth. And variants are specific genes that are histones, but they actually have specific structural modifications, and therefore we call them variants. Then there's the DNA methylation patterns that are very important as well in enhancers. And for our purposes, the most important part at the end of the day is to identify these regions and then nail down to which transcription factors can be regulating such genes, and then associate those transcription factors to potentially drivers of tumorigenesis. And something that I'm not going to be talking so much about today is non-coding RNAs, which are also expressed in these elements. So one thing that basically turned us to apply some of the novel sequencing technologies available was basically because at Mayo Clinic we have so much translational scientists, and we have a very good access to patient samples. We really wanted to start using epigenomic analysis that could allow us to do a lot of look at a lot of different samples, in a cost-effective manner. And so ATAC-seq is one of these platforms. I don't know if you know much about it, but all you need to know about ATAC-seq is that basically it makes uses of a transposase. This transposase has been modified molecularly. It's called TN5, and it's hyperactive. So if you put it in a cell in the nucleus, it will basically bind random places in the genome. But because the way we actually apply it to a nucleus over the course of 30 minutes, for example, we basically only give this transposase access to the regions that are open. Those open chromatin regions, that we call them, are basically the enhancers and promoters, because that's where nucleosomes are devoid, and that's pretty much because of transcription factors binding to those regions. So the way by looking at the way basically the genome is breathing this open chromatin regions, we can actually start assessing different features of the epigenome, such as open chromatin regions. We can cluster these together and try to identify patterns, and we can eventually look at transcription factors that bind to them by doing bioinformatic analysis. And as we understand from talks also from the previous session, it's actually now very important to also understand the tumor heterogeneity. And because now we understand that tumors are not all the same. When we treat a tumor with a particular chemotherapy or a specific agent, we already know that the majority of the time some of the cells will actually be resistant to this. And whether you want to call them drug-resistant cells, cancer stem cells, or other tumor-initiating cells, at the end of the day, we actually have been really struggling to identify patterns from these cells that could really explain why these cells are not going to be killed. And so in our approach, looking at the epigenome, is really to try to understand if this extra layer of understanding will allow us to define better, identify the cells that are not going to be basically targeted. And this is what we call basically transcriptional dependencies. We're trying to find transcription factors that are associated with the specific patterns that could potentially be targeted. And so to do that, in my lab, we've been using this single-cell approach. It's called the Multiome. It's available from 10x Genomics. And it basically makes use of single nuclear that we perform ATAC-seq and also isolate the RNA. And so you can already appreciate that by integrating these two approaches, epigenomic and transcriptomic, you can start understanding a little bit better of what could be happening to these cells. And this is just in the bottom, just a feature from the 10x Genomics showing that basically what we're actually doing is isolating RNA from the nucleus. That's an important feature here, and that's actually something that we try to understand by comparing single-cell RNA-seq and single-cell nuclear-seq. And so the first thing that we actually did when we started applying this technology was basically to compare from similar patient samples. In this case, it was PBMCs. We were comparing single-cell RNA-seq to our multiome approach, and in this case, we isolate only the transcript. So we're looking at the single nuclear RNA-seq, and as you can see, you probably don't see very well, but this approach really allows us to characterize cell types using these two technologies, and overall, we really didn't see many differences. But we did see, obviously, some differences, and this is also true, for example, when we compare RNA and protein. And what was very striking was that there is a large group of genes that don't really correlate very well between the two platforms. But when we actually compare the expression of these genes that don't pair very well, we actually see that the majority of the negative correlated genes are poorly expressed. And so we believe that this is really a technical issue. If you increase the ability to get coverage of these genes, you will probably lose some of those negative correlated genes. And so the truth is also, this is very important as well, because the marker genes, the genes that we typically use to identify cell types, they're all positive correlated, and that's very good to see. So let's get to some molecular mechanisms of these enhancers, and this is something that in my lab we've been working on. This is work by my computational biologist Wazim and Amelie in the lab. So we basically were very interested in these patterns, as I mentioned, of enhancers. I'm not going to really go into the bioinformatics of like how we basically came up to this, came down to this analysis, but what we basically defined was 5 different subgroups of regulatory elements. We call those the ATAC only, so this is just open chromatin regions. There's the inactive that are typically marked by repressive histone marks, the active that are, the ones that we typically look at in some of these approaches. Then there's the regulatory elements that, are really close to the promoters, so we call them promoter-like. And then there's the new subclass that we call macro-H2A or macro-bound enhancers. So these macrobound enhancers have a repressive effect globally, and what we actually believe they're doing is basically preventing some of the oncogenic pathways to be reactivated in the case of cancer. So one example that we actually did was by looking at the reactivation of macrobound enhancers that had been identified in normal cells, in non-malignant cells, and if you look at the reactivation of these enhancers in breast cancer, in a variety of different breast cancer cell types, you can actually appreciate that if you do PCA, you see that the reactivation of these enhancers pretty much marks their subtype, which is kind of an interesting way to see that indeed this regulatory network that has been protected in normal cells seem to have a very important and cell-specific impact in breast cancer. They also are associated with mutations or associated variants. But very interestingly, these macrobound enhancers, this repressive environment, really associates with transcription factors very specifically. Not all transcription factors are being affected by these macrobound enhancers. And so we basically took a breast cancer cell line, in this case is a luminal cell, that it's estrogen responsive, and we looked at where the macrobound enhancers are regulating in these particular cell types. And we saw that, very importantly, these macrobound enhancers, even though they have a repressive effect, they actually tend to be enriched in genes, in transcription factors that are associated with estrogen response. And so what we did molecularly was we knock out macro-H2A in this cell line, and we were actually indeed were able to confirm that by removing these enhancers, you were able to increase the estrogen response in these cells. And then we did single-cell ATAC-seq in these specific cells and compared the knockout with the parental MCF7 cells. And indeed, again, we see that the ATAC-seq of the open chromatin regions are pointing out to enrichment in transcription factors that are associated with estrogen. A lot of these are actually specific to the macrobound enhancers, so even though we were not sure if removing the macro-H2A would only affect enhancers, we actually see that for the most part, that's what we actually see. And these are pretty complex diagrams, but I wanted to focus on the sister interactions, the circus plot, where you actually see a comparison between what is most likely to interact with these macrobound enhancers in different colors. And so the macrobound enhancers are in blue, and the gain interactions are the darker blue. So you can see that there's a lot of blue in there, meaning that a lot of the enhancers that were not active in the parental cell now are becoming enriched. We then actually looked at the normal stage, and so we make use of mammary epithelial cells that come from a knockout mouse. In this case, it's a double knockout because there's 2 genes for macro-H2A, and we actually see that there's an increase in stem cell activity in these knockouts, and so we believe that macro-H2A again is preventing the cells from mechanisms that are associated with stem cell differentiation. We then did use this platform that I was talking about, the multi-omic platform, to really start narrowing down what are the transcription factors or chromatin remodelers that are involved in this process. And we actually were able to find that, for example, BRD4, which is a reader for enhancers, was highly affected. So in the knockout, we actually see an increase of BRD4 binding to all these enhancers that we had annotated. And so basically, to make a very long story short, we're really trying to now define the mechanisms that are underlying these this novel regulatory pathways. And so we believe that these macrobond enhancers that, by the way, have this large domain that I'm depicting there, they are actually able to prevent the binding from either BRD4 directly or through some of these partners such as Zim and Date, or actually the histone marks themselves, such as H4K12 acetylation, which is read by some of these partners. And overall, what we actually believe is that this process really allows the cells to have a good way to fine-tune the expression of specific genes. And so what are we doing now with some of these technologies in the lab? So here I'm actually putting out 4 different examples of projects that we have. We are now focusing quite a bit in ovarian cancer. And we have been actually actively collecting samples that have come directly from debulking surgeries. And some of our interests are obviously to look at the tumor microenvironment and to try to understand some of these interactions that potentially could be preventative of immunotherapies. And in this case, because by using multiome, we're not only able to identify which cell types and which genes are downregulated, but we can try to go take a step back and understand maybe there are some transcription factors that could be targeted in such a way. And an important part of what we're actually trying to do is actually driven by our interest in identifying these cells that are basically resistant to drugs. And so cancer stem cells are some of the important signatures that we're really trying to identify using these methodologies. And one of my students is actually trying to look at the dark matter of the genome and actually is trying to identify mechanisms of activation of transposable elements, which are actually very important during tumor formation. But one example that I want to just show you, and this is something that we're going to submit very soon, is about clonal hematopoiesis, and this is actually a project that started during the pandemic. So in clonal hematopoiesis, as you probably are aware, this is an aging-related disease, and there's an accumulation of mutations that basically seem to give competitive fitness to hematopoietic stem cells. And what is interesting, at least from my standpoint, is that the top mutations from these clonal hematopoiesis of indeterminate potential are DNMT3A and TET2, which are both affecting DNA methylation, but they actually have opposing effects. And so even though we're talking about a specific phenotype that it's very commonly seen in the clinic, we actually we are having a hard time understanding why 2 opposing genes have pretty much the same outcome. And so DNMT3A is a writer of DNA methylation, so it adds methylation to the DNA, and TET2 is functionally an eraser of DNA methylation. So what we did during the pandemic was we were able to start collecting patient samples from the clinic and basically annotating whether they had DNMT3A or TET2 mutations. And because most of the people that were getting highly affected were older, we were actually able to detect quite a few of them. And we did a panel of assays just to kind of like really try to understand, give a multi-omic approach to try to understand what we could, how these 2 mutations could be so different. And so we did some of these features, platform. We used some of the platforms that are singled out here. Tapestry basically allows us to look at using cell surface markers to identify the cell types, and then with a panel of genes we can actually detect the mutations, so we can identify the actual cells that have the mutations in them. We used the multi-omic platform that I already mentioned, and we also did Gotcha, which is a very novel approach that is really exciting to us, where you can actually use single-cell latex-seq and then also using probes detect the genotype from these cells. So again, we can look at open chromatin and also determine whether the mutation is present or not. We also did DNA methylation because, as I mentioned, this is affects the methylation. But in this case, we still haven't really gotten to the point where we can actually do this at a single cell level. So this is a very good collaboration with Dr. Pat Naik. He's also at Mayo Clinic and is the director of the Epigenomics program, and with Moritz Binder and some people in my lab. And where we actually were able to determine from the beginning was that even though, again, CHIP is globally seen as globally with these two different mutations, we actually saw a very striking differences between the impact of DNMT3A versus TET2. And so we believe that DNMT3A in this case really affects the patient outcome for the COVID patients. And this is a very busy slide, but this is really just kind of like summing up everything, all the platforms that I was actually talking about. So on the top, you can actually see all these little interactions, and so that's data that we collect from the epigenome, and you can see that the TET2 mutants have very very few interactions, and this is around the gene that we identified as a potential biomarker for severity. It's IL-32, it's a cytokine, and we believe that for DNMT3A patients, it's highly active, and as you can see by ATAC-seq, there's a lot of interactions around this gene. Now, what we can actually then trace back is to look at all these interactions and try to identify specific transcription factors that could be driving this activation. But also, we were able to determine that some of these enhancers were associated with methylation patterns that were basically lost upon DNMT3A mutation, specifically in cells that were mutated. So by basically using gotcha, we were able to see that the open chromatin region associated with some of these binding sites were actually specifically high in the cells that are mutated and not in the cells that were not mutated. So this is kind of like a very exciting single cell resolution platform. So just to summarize, we've basically been looking at the epigenome at the single cell level. We identified this novel class of enhancers that we believe can be very interesting in the context of cancer. And we've been using this multi-omic approach to really understand transcription factors in disease. I just want to finish by acknowledging, of course, my group, my small group, and also some of the collaborators that I mentioned along the way, such as Rinal Patnaik. One important thing that you probably want to know is that this multi-omic platform is obviously available through 10x Genomics, but at Mayo Clinic we also have a recharge facility that allows other researchers to actually use it. And so if you're interested in this technique, you can reach out to us, and we can help you with getting your experiments forward, and looking forward to your questions. Thank you.
It is my great pleasure now to introduce the next speaker, Dr. Kelley, who is a gastrointestinal oncologist who specializes in cancers arising from the liver, including HCC, bile duct cancer, and gallbladder carcinoma. Your research interests include developing clinical trials of new treatments and combination therapies for this cancer, including immunotherapy approaches, as well as translational research to identify molecular and genetic biomarkers of response. Thank you for being here, Kelley. It is really my pleasure to have you here, and her talk is entitled Germline Genetics and Risk of Cholangiocarcinoma. Welcome.
Well, thanks so much to Daniela and Sumera and CCF for the honor of speaking today in this session. Here are my disclosures. I'm a clinical trialist, so I do have disclosures related to conduct and development of clinical trials. And another disclosure is that I'm a clinical researcher, not a lab-based or basic science researcher, so it is not without some imposter syndrome that I stand here in a room of scientists to hopefully present something that is intriguing and may guide some interest in these pathways as I present the prevalence and some of what we do know about germline risk in cholangiocarcinoma. So here's an outline of the talk. I'll focus on biliary tract cancers in general, and of course, cholangiocarcinoma, or CCA, comprises the majority of the data I'll show, and start with where these fit in our known collection of hereditary defined cancer syndromes, and then delve into the prevalence of pathogenic germline aberrations, or PGA. That's an acronym I'll use a lot, PGA, in biliary tract cancers, with a specific focus on those associated with DNA damage repair and homologous recombination deficiency, or HRD. And then last, turn to the clinical implications of these findings. So beginning with hereditary cancer syndromes and BTC. Here is our obligatory background slide placing BTC in the context of its complex anatomy. And this is really in part intended to show, here we go. show the acronyms I'll be using: CCA for cholangiocarcinoma, GBC for gallbladder, of course, and NGS for next-generation sequencing. Beyond the anatomic complexity of biliary tract cancers, the heterogeneous biology is another added layer of enormous intricacy, as Rory showed us so elegantly in the last session. And as we increasingly are increasingly testing patients for somatic mutations in the clinic to guide our treatment decisions, we're realizing that there are candidate germline aberrations popping up on our next-gen sequencing panels, and it's really incumbent upon us to understand who and where are these showing up, and how do we follow them up. So there are a number of hereditary cancer syndromes with high penetrance that do increase the risk for biliary tract cancers. And the best known of these are the DNA damage response, or DDR deficiency family of genes, including Lynch syndrome or mismatch repair deficiency. And in a cohort of patients defined by presence of colorectal cancer and mismatch repair deficiency, the subsequent risk of cholangiocarcinoma or biliary tract cancer in general in those patients is about 1% in 10 years, which is much, much higher than the population risk. So that's the MMR increased risk. Now, hereditary breast and ovarian cancer, also homologous recombination repair deficiency genes, have also been shown to be associated with an increased risk of biliary tract cancers. And in the right on table 1 shows very old data from one of the early breast cancer linkage consortium studies showing a much higher than expected risk of gallbladder and bile duct cancers in patients with a known BRCA2 mutation. And though the numbers are quite small for individual patients, the expected frequency was, the incidence observed in this cohort was much, much higher than the expected frequency in the normal population, leading to statistical significance and approximately a 5-fold increased risk for biliary tract cancer in BRCA2 carriers. Now, there's also an increased risk in the BAP1 tumor predisposition syndrome, which is another high penetrance germline hereditary cancer syndrome, which I'll show on the next slide, related to cell cycle signaling pathways. But a caveat is that determining these associations is certainly challenged by the rarity of biliary tract cancers and the individual syndromes. So there may certainly be others that are not well described to date. So I'd like to focus a little bit more on DNA damage response, or DDR. This is a complex family of aberrations related to DNA repair mechanisms, and include the best known ones shown here. And so these include mismatch repair deficiency, which cause insertions and deletions, and frame shift mutagenesis that we know as mismatch repair deficiency. This is an old slide, so it doesn't include all the potential genes and tumor types, but colorectal cancer, of course, is a classic cancer associated with mismatch repair deficiency in a subset. Also, homologous recombination deficiency derives from double-stranded DNA breaks with a host of genes that can be associated with HRD and subsequent tumor phenotypes shown here. And a key point is that pathogenicity in DNA damage response mutations is really thought to require a loss of heterozygosity, or LOH, meaning a second somatic hit in the previously wild type or normal allele. And that's not always easy to detect because it can be due to epigenetics, or also chromosomal loss. Things are not captured necessarily in a clinical panel. So turning to another important known hereditary cancer syndrome, which has been shown to be associated with the development of biliary tract cancers, is BRCA-associated protein, or BAP1 tumor predisposition syndrome. This is a rather pleiotropic deubiquitinating enzyme involved in a variety of cell activities, including cell cycle signaling and DDR. It acts as a tumor suppressor. And germline deficiency has a high penetrance for multiple different cancers, particularly mesothelioma, but also cholangiocarcinoma. And shown on the right are actually somatic mutations, or rather, tumor mutations that could be a mix of somatic and germline here from a Foundation 1 database that showed that cholangiocarcinoma came in second after mesothelioma for incidence of inactivating BAP1 mutations, underscoring the importance of BAP1 and cholangiocarcinoma as a potential germline cause. And we know that also in probands with a familial BAP1 mutation, cholangiocarcinoma occurs at a much higher than population expected risk, about 1.4%. So there are guidelines for genetic counseling, testing, and risk reduction measures in patients with known germline BAP1. So to summarize this point, there are several well-known hereditary cancer syndromes that also are associated with an increased risk of biliary tract cancers, and these include Lynch and MMR, homologous recombination deficiencies, or HRD, and BAP1. There are probably others that are not well described owing to rarity of both the mutation as well as biliary tract cancers. These hereditary cancer mutations are fundamental to tumor biology and impact prognosis and treatment response, as well as the risk of developing the cancer. So turning to the next point is, as a clinician, when I see a patient with a diagnosis of biliary tract cancer, what is the likelihood that that patient has a germline mutation that we should be testing for, and particularly HRD? So this is really the take-home slide for this point of the talk, where I've really summarized a host of cohorts that include a meaningful number of biliary tract cancer patients and include at least a multi-gene panel germline testing, and some also included paired tumor testing. So these are germline multi-gene panel biliary tract cancer cohorts across a globally representative set of studies. And again, the take-home points here are that overall pathogenic germline aberrations, again, that's the PGA acronym used here, occur in a meaningful number of biliary tract cancer patients, up to 16%. And these are most common in DDR pathways. HRD germline mutations are present in around 3 to 6%, and germline BRCA1/2 in 1 to 5% across these large studies. So these are meaningful numbers when you're talking about high penetrance germline aberrations as a clinician sitting across from that patient. And there is a trend across these studies that, as one might expect, that these pathogenic germline aberrations are more common in younger patients with a positive family history, and actually in Asian patients as well, for unclear reasons. And there was no consistent difference across studies so far between intrahepatic, distal, and perihilar and gallbladder location. So anatomic location, to Rory's point, doesn't necessarily confer risk one way or the other. So I've selected out a couple of these studies for the interesting points that they raise. One is this Tianjin Chinese cohort, which is a relatively large cohort out of Tianjin Medical University with paired tumor normal NGS, and they showed pathogenic germline aberrations in about 10%, trended towards younger age. And in the heat map here, we can see that there was really no clear difference between the anatomic subsite. Light green, pink, and blue are the different anatomic subsites. And we can see that the most prevalent mutations were in DDR genes, BRCA in about 1.3, and then here's the HRD genes in about 5%. So again, not a trivial number, particularly for HRD. Also interesting from this cohort, I found the green bars here show patients with a family history compared to patients without a family history. And we can see that patients were much more likely to have a pathogenic or likely pathogenic germline aberration if they also had a first degree or family history positivity. Another interesting finding from this study was shown here on the right with these pink and purple bars. They took the same exact genes in the Tianjin cohort and compared it to C-Bioportal in a Western cohort, and showed a higher prevalence of DDR pathogenic germline aberrations in the Chinese or pink bars compared to patients from the Western cohort. So intriguing finding, suggesting higher rates in Chinese or Asian patients. So this is a different cohort from Tempus with about 840 patients with biliary tract cancers out of a much larger deep dive into over almost 35,000 patients across solid tumors. So in the 840 patients with cholangiocarcinoma, predominantly Caucasian, paired tumor normal NGS was performed, and a smaller proportion of patients had PGAs, about 4.3%. 1.2% had BRCA1/2, and this is an interesting point I wanted to call out, that of those patients, about a third of those with BRCA1/2 had a second hit, so they were truly HRD, and when they did the paired tumor and normal. What they show, the way Tim Yap and his co-authors presented this, is showing on the bottom graph here are tumors with an established guideline for genetic testing, like breast, pancreas, prostate, et cetera, where we have guidelines to actually test these tumors for germline mutations. And we see the numbers and incidence of these pathogenic aberrations is not dissimilar from tumors like cholangio, which don't yet have a guideline. So this tells us we're missing out on having a formal guideline. So a last cohort I wanted to highlight is a large Biobank Japan and Hokkaido cohort, which looked at over 1,200 cases of biliary tract cancer in Japan. and compared the incidence of germline aberrations to over 37,000 controls without cancer and without family history. And what they showed was that about over 5% of the biliary tract cancers compared to 1% of the controls had a pathogenic germline aberration, supporting that these are causal. And they showed significance for BRCA1, BRCA2, and MSH6, reinforcing that mismatch repair and HRD through BRCA1 and 2 are causal for biliary tract cancers. The age was slightly younger, and there was an increased family history. So again, these are fitting what we know, but data to support that we should be doing testing in these patients. And an interesting deep dive they took in about 45 of the patients in this cohort was that they performed tumor WGS in addition to their germline testing, and showed that really a second hit was required to see an HRD signature, as we'd expect, but really reinforces the importance that we should be looking for this to know which patients are truly HRD. So limitations of these types of cohorts are that there's heterogeneity in testing, there's varying gene lists and coverage across panels, we can be missing large genomic rearrangements. The source of DNA, having paired tumor versus paired normal, skews our findings, and low tumor purity can also confound assessment of allele fraction. And then just some really important clinical biases to be aware of, that we do introduce selection and referral biases in that retrospective cohorts do tend to select for high-risk, younger, positive patients based on who we refer for testing. There's ascertainment bias for things like family history and a poor prognosis cancer, where we may not actually have a healthy enough population to test. And also, really importantly, regional race and ethnicity variations require that our panels and probes be validated in broad populations, because particularly the LGRs can be different. So summary for this section about prevalence, again, the take-home message is around 4 to 16%, so meaningful number when we look at that 16% upper estimate of biliary tract cancer patients do harbor pathogenic germline aberrations, and most of these are DDR pathways. The HRD mutations may be as high as 3 to 6% in the germline context. BRCA1, 2, 1 to 5% across these heterogeneous studies, and we expect, we do see some enrichment in younger positive family history and Asian cohorts. No clear-cut differences by anatomic subsite, and again, bear in mind the selection and ascertainment biases, including testing modality. So lastly, as an oncologist who's thinking about how to treat this patient when I see someone with a germline mutation potentially, on high in cancer, we know that there are pan-cancer clinical implications of pathogenic germline aberrations, and particularly for genes with high penetrance in germline carriers. We know that having a germline mutation in BRCA1, 2, for example, is the classic example, confers an enormous risk, lifetime risk of cancer, greater than 60% for ovarian, and very high for pancreas and other cancers as well. Sorry, breast and ovarian, and also pancreas and others. And so finding a mutation like this makes it incumbent upon us to refer that patient and also their family members for counseling and consideration of testing, as well as potentially risk reduction measures such as prophylactic mastectomy or oophorectomy. And so that's one of the reasons why we need to know about these, is for the patient and their family members. But again, speaking as the oncologist, we also assume that we know that these mutations impact tumor biology and could be guiding our choice of therapies. So in particular, we know that we already have an abundance of data across tumor types, including cholangiocarcinoma, that immune checkpoint inhibitors have a substantially higher response in patients with mismatch repair deficiency or microsatellite instability, and that has led to a tumor-agnostic FDA approval. We have less data so far about platinum chemotherapy in biliary tract cancer, though we know that HRD is a predictor for response in other tumor types, so we assume it should be in biliary. We just don't have that much data yet. And likewise for PARP inhibitors, we have data in other tumor types, but not much in biliary. So I'd like to highlight some of the unanswered questions. We don't yet know how to use DDR, HRD, and BAP1 mutations to predict treatment response in biliary tract cancers. We can infer from other tumor types. And we also do not yet have established guidelines for referral for germline testing and screening. So, oops, sorry about the bad animations there. So focusing first on the platinum question, there are some retrospective case series that suggest that platinum-based therapy may have better outcomes in patients with DDR mutations. but this is really retrospective. A Korean study shown with the Kaplan-Meiers with over 88 patients showed that there was longer PFS and OS in patients harboring a DDR mutation than those without. Which is, again, what we expect from other tumor types. But Angela Lamarca, one of the conference chairs this year, looked at this question in ABC06, which looked at Fulfax, a combination with oxaliplatin, a different platinum generally, treated in the second line in biliary tract cancer, and despite adjusting for prognostic covariates, did not see a clear-cut difference in outcomes in the ABC06 trial. So we have a little bit of mixed data in these, specifically looking at platinum-based therapy. But I think I'd like to show now the best prospective data we have right now, which is from the TOPAZ-1 first-line clinical trial looking at gemcitabine cisplatin chemotherapy, so platinum being the cisplatin, combined with an immune checkpoint inhibitor in first-line biliary tract cancer. And from the TOPAZ-1 study, we saw that there were, as expected, a meaningful number of BRCA1 and 2 and ATM mutation patients in particular that were in the sort of the top subset of genomic aberrations overall. Now, I'll just the caveat, this is a mix of tumor and of somatic and germline mutations because this is based on tumor testing. But what they showed in these patients in TOPAZ-1 is that the BRCA1 and 2 biliary tract cancer patients had a higher objective response rate in both the control arm with just GemCis alone, shown here in this yellow bar, as well as the GemCis plus durvalumab immune checkpoint arm, shown here in purple. So this BRCA1/2 group did really better than all the other tumors when we look at the objective response rate, and PFS and OS were similar. They trended a little bit better as well in small sample sizes. So this is intriguing. And we also know from some clinical cohorts that there's a higher TMB in these patients, so it reinforces there could also be a trend towards better response to immune checkpoint inhibitor. So right now, we think that these patients do well with the TOPAZ-1 GemCis plus Durva regimen. What about PARP inhibitors? We have really no prospective data to date. There's case series suggesting some individual patients with prolonged responses. But a huge challenge in what we see in the retrospective data is lack of accounting for whether these are loss of heterozygosity or just a single hit type patients. This table shows a list of ongoing trials in both HRD selected and unselected designs. And I think, again, the big caveat is unless they're really checking paired normal, we are not going to know if these are HRD patients or true LOH or not. And there's one small study that showed an intriguing high rate of response or stabilization to PARP inhibition in BAP1 as well, just as an aside. So right now, there are no specific biliary tract cancer guidelines for which patients we should be testing for germline pathogenic aberrations, but the incidence, as I showed earlier, is quite similar to what we expect in other tumor types that do have an indication for germline testing. And I think it's really important for us to remember that those pathogenic germline aberrations that I've just shown do occur in biliary tract cancer are ones that, if you know about it, there are risk mitigation strategies, family testing, and management plans. So it really speaks to the point that we should be testing these patients if we have an index of suspicion. And just the point about this is not only to guide us in the clinic, but we need these guidelines to support reimbursement coverage decisions because that's the biggest limitation, not just the physician ordering these tests. The NCCN guidelines do not have any specific guidelines for biliary tract cancer. They do for breast, ovarian, pancreas, prostate, and I would submit we should really lobby for biliary based on the data we have. Even though we don't have a drug, we have these high-risk, high-penetrant mutations we should find. There is no clear role if we do find these for biliary tract cancer screening yet, but I think that's a topic of interest in pancreas and biliary tract cancer screening clinics for these germline mutation carriers. That's all I have on this topic today, but I'm hoping that this will be a topic for a future session or down the road in a couple of years. And then just to summarize and conclude, the certain pathogenic germline aberrations do have therapeutic implications in advanced biliary tract cancers, potentially a better outcome on platinum-based therapy and better outcomes based on response rate to TOPAZ-1, GemCis plus Durva for the BRCA1 and 2 patients in particular. And we look to the prospective PARP trials ongoing. These high penetrance mutations warrant counseling and consideration of risk reduction measures in your patients when you find them. And the key points for future directions, I've just talked about testing, but I think this is a key point that I'm interested in. As I mentioned, I'm a clinical trialist, and we really need to develop more trials of PARP inhibitor-based therapies and other DDR and HRD targeted strategies for this really meaningful subset of patients. And when we do so, we need to include tumor and normal testing, because otherwise we could be missing a signal if patients are not true HRD and we're just testing one or the other. And this is my last point, is that I refer my patients with biliary tract cancers for germline testing and submit papers and appeal whenever I need to, if their age is less than 50, if they have a positive family history, or if there's a candidate germline mutation on tumor testing. And I should say positive family history or personal history. So if that patient also had a breast cancer themselves 10 years earlier, I will refer them for that reason as well. And we need these clinical guidelines so that we can support reimbursement more easily across the board. So with that, I'll thank you for your attention and for the honor of this invitation.
All right, thank you, Katie, for a wonderful talk. Our next speaker is Daniela, who you've already met several times before, and many of you already know. Dr. Sia is assistant professor at Mount Sinai in the Department of Medicine. She's a cancer biologist with a real commitment to cholangiocarcinoma research, has made significant contributions to our understanding of the genomics of this tumor type, tumor immunology, fusion proteins, the list goes on. And today she's going to talk to us about genomics and subtype.
Thank you, Ben, for the nice introduction. It is my pleasure to be here. I'm really happy to have had the opportunity to put together this program with Sumera, and I hope you're enjoying the discussions, and we will have more later. While I figured out how this works, because it has changed. Yes. So I would say that this is the only conference where I do not need to introduce cholangiocarcinoma, which is great, so I can skip the first slide. But we have heard already this morning about the importance of genetics. So I want to focus more on the heterogeneity of cholangiocarcinoma, which we know that is one of the main challenges. And if you look just here, there is a long list when it comes to heterogeneity of cholangiocarcinoma. Many aspects are different. We have heard already about the location, how there are different subtypes, introducing also risk factor, histopathology, how these tumors look. But how we learn more, and we realize that we know really little still. We know also the tumor microenvironment is different, also related to the different location, but how we will focus here also genetic alterations are different, which is puzzling because again, it keeps opening question, is there any relationship with different cell of origin? We have heard this morning differences between intrahepatic and extrahepatic. What is the cause? What is the chicken or the egg? And this is what we're trying to dissect, that we have tried for a long time with genomics, right? You know that the application of next generation sequencing has been one of the biggest revolutions for cholangiocarcinoma, but we're always quite behind. There is always a lag. We have heard already that unfortunately, with the variety of the tumors and the difficulties in getting access to tissues, we're always kind of behind compared to other cancers. And I wanted just to introduce here, if you look here when it happened, I cannot read, I'm sorry, I'm getting old. Maybe you can read better than me, but it's like 2010 when we had already genome sequencing of several cancers, but still not cholangiocarcinoma. We're always behind. And interesting, I found one of the first reviews that I ever wrote in cholangiocarcinoma was 2013. And if you look at this list, this is the list of genetic alteration that we knew back then. And there were like 5 or 6 genes, the first rows. So it's kind of interesting that in 2024, a little bit over 10 years, how much we have learned. And I found really interesting that at the end of this review, we actually say that we hoped that with the new sequencing technologies, we will learn more about the genomics of cholangiocarcinoma. And again, as you know, this happened. And this is one beautiful review from Juan in Cancer Discovery 2017. And you can see that now we go way beyond the 5 genetic alterations that we knew in 2013. And not only we know many more, but also we know that they are not always the same across the different subtypes. So I go back to what was mentioned before, that location is important, but also it's not. And we need to go beyond the differences intrahepatic versus extrahepatic, or hilar and perihilar. And interesting, as I say, this is a great evolution because, of course, he brought precision oncology to cholangiocarcinoma. As you know, HCC and cholangiocarcinoma both arise within the liver, right? The microenvironment seems to be more similar than when we compare to extrahepatic. But interested in HCC, almost none of the alterations are targetable, whereas with cholangiocarcinoma, application of next-generation sequencing has brought important changes into the management with FGFR inhibitors and IDH inhibitors that we are all familiar with. So, of course, when it comes to cholangiocarcinoma, as I said, in the past 10 years, we have learned a great deal thanks to the application of whole exome sequencing and whole genome sequencing. And we know way more about intrahepatic cholangiocarcinoma than the other subtypes when we refer to them in terms of location. This, for example, has been mentioned before. This is a beautiful study published in Cancer Cell 2022. This is a large cohort from China, more than 250 intrahepatic cholangiocarcinoma. And these studies have helped a great deal in understanding the main drivers. And you're all familiar with them, KRAS and TP53. We've all heard about them. But the most important is that there is heterogeneity when it comes to intrahepatic cholangiocarcinoma, besides the location, and besides the anatomical location, also if we think about geographical location. And in this study, for example, they compared, this is the Fudan, the first in red in this graph, when they compared to the results of genetic alteration found in the cohort from Sloan Kettering and the ICGC, which is mostly from Singapore and Thailand, there were important differences. For example, TP53 and KRAS seems to be more frequent in Southeast Asia, whereas when it comes to IDH1, and we will see later FGFR2 fusions, this seems to be more frequent in Western countries. And of course, when we have to explain these differences, the main things that come to mind is different etiological factors. Here is just to report that the FGFR2 fusion were also identified in this cohort from China, and they were around 10%. FGFR2 fusion, you've heard about them, they're quite interesting. These were identified by RNA-seq in 2014 for the first time, and now we know that we have more than 60 partners. FGFR2 can due to breakup points in the chromosome, within the same chromosome, between different chromosomes, we have the two genes get fused together and they form this oncogenic form of FGFR2, which is always active and signaling within the cells. FGFR2 fusion are the most relevant, also the most frequent, around 20%, although there are quite important differences in terms of incidence, but they're not the only mutations or alterations that impact FGFR. We have also amplification that induce expression, but we also have FGFR2 activated mutations, although these are quite less frequent than fusions, where indeed less than 5%. More recently, also, deletion of the extracellular domain and free deletion have been reported and seems to have a similar effect to the fusion. They all seem to activate FGFR2, suggesting that all these patients could be responsive to FGFR inhibitors. However, as I say, there are important differences in terms of location, and one of the easiest way to explain this is risk factors. So we're familiar, for example, that in Asia, in Southeast Asia, liver fluke are quite frequent, whereas we don't see them here in Western countries. And in this beautiful study in cancer discovery, actually, this is one of the largest cohorts where they were able to compare liver fluke positive versus liver fluke negative. And this is all kinase KRAS-related genes. And interesting FGFR2 fusion, they were quite frequent in liver fluke negative, but they were almost absent or very rare liver fluke positive. And this was confirmed in these follow-up studies, and you can see the numbers. Those with liver fluke positive have very low frequency of FGFR2 fusions. They went beyond, and in the initial cancer discovery, actually, by comparing these large numbers of liver fluke positive versus negative, they identified 32 different genes that were enriched. And I'd like to highlight, for example, BAP1, and we will see later why, but also using this large cohorts, they were able to identify novel tumor suppressors. And I just want to give the example of this RAS-A1, which is a MAP kinase-related gene, and its deletion actually induce more aggressive tumors. As I say, there is importance in the difference according to the different etiology, and they went beyond. And the Yusaku et al. Actually looked also at the epigenome. We have heard about the importance of epigenetics before. And interesting, when they performed this analysis of epigenetics across all these cancers, they observed that not only the genetics was different. I say, for example, BAP1 was more enriched in liver fluke negative, along with IDH, but also the epigenetics was different, suggesting that indeed maybe the tumor genetic process is not the same. And heterogeneity, for example, here I just want to highlight cluster 1, the blue on the left, and cluster 4 on the right. They're completely the opposite extreme. And you can see the liver fluke positive are enriched in cluster 1, whereas the liver fluke negative are enriched in cluster 4. And genetics follow. These differences are along. So we need kind of integrate epigenetics and genomics to understand. And interesting, when in cluster 4 there is an enrichment of IDH, we know the IDH 1 and 2 have been associated with hypermethylation. And indeed, there was difference since sure there was a hypermethylation of the shores, whereas in cluster 1 there was island hypermethylation, suggesting again this link between etiology, genetics, and epigenetics. Again, we have heard about subtypes. Just looking at intrahepatic cholangiocarcinoma, there is important heterogeneity. We know that there are two main subtypes called small type and large type, where large type seems to be occurring in large intrahepatic bile duct. It seems to be more similar to what occurs outside of the liver. And interesting, when they've been compared in terms of molecular genetics, also the large type seems to be more similar to what we call extrahepatic, with more KRAS and TP53 mutations, whereas the ones with small type are enriched in IDH and FGFR2 fusions. Going beyond, again, let's go back to the anatomical location. We have even more heterogeneity if we compare intrahepatic cholangiocarcinoma to perihilar, distal, and gallbladder. This is a nice study from Wardell Journal of Hepatology 2018, really large cohort, although as I said, unfortunately, extrahepatic we always have less, although it's more frequent, so it's kind of a contradiction. I think it just to highlight how more work needs to be done in terms of genetics when it comes to these more rare subtypes. And interesting, they observe differences when comparing intrahepatic versus how occurs outside of the liver. And as you can see here, TP53, KRAS, SMAD4, and LBB3 were more frequent in extrahepatic. Interesting, when it comes to the two forms of extrahepatic cholangiocarcinoma, in our study in Journal of Hepatology 2021, where we had 189 extrahepatic cholangiocarcinoma, including both distal and perihilar, we did not observe significant differences between the two. But again, I think that we need more samples. This was the largest cohort, but as I said, we need to do more work. Interesting, there are more studies kind of focus on understanding these differences in terms of genetics. This is one study recently published in Nature Communication when they compared perihilar to intrahepatic cholangiocarcinoma. There are important structural differences that go beyond genetics. Overall, there are more single nucleotide variations in intrahepatic, but also differences in terms of chromosomal aberrations. And in particular here, they can identify a specific driver that seems to be enriched more in one subtype than the other. Why they're important? Well, we heard about the impact on management, right? But again, what we are not doing is an integrated approach. I've heard before from Rory, right, suggesting that we need to go beyond the single levels. We need to kind of put together all the information. And I like this, although simplistic, the way we need to integrate all the information that we're collecting here and there. Type of intrahepatic cholangiocarcinoma, but also genetics, risk factors, everything needs to be combined together to kind of identify the best stratification that goes beyond anatomical location, geographical location, and risk factors. Everything needs to be integrated to better understand how to handle these really challenging tumors. And of course, these alterations dictate response. The case of FGFR2 fusion, they respond to FGFR inhibitors, IDH with IDH inhibitors, but they also dictate resistance. And this is the very elegant case of FGFR, where patients, after an initial response, they develop acquired resistance mutation in the FGFR kinase. And of course, this is important to understand this, because unfortunately, albeit successful, we know the patients progress quite quickly on FGFR inhibitors, and we're just now trying to understand how to move forward and prolong the survival and the responses. We have heard about immunotherapy. We know that now it's integrated in the new standard of care, and genetics is also important. And we've heard also from Katie before, and I look forward to discuss during the panel discussion. This is the TOPAZ trial that brought immunotherapy into the management of cholangiocarcinoma. We know that a single agent, unfortunately, are quite unsuccessful. What about genetics? And as we know, in the case in other cancer, expression of PD-L1, just expression of PD-L1 has been associated with response, not in cholangiocarcinoma, where the only predictor so far has been presence of mismatch repair deficiencies. Unfortunately, these, as you can see, are quite rare. We have 2-3%. When it comes to genetic alteration, we are just really starting to understand the connection between tumor and the rest I refer to immune and stroma. So far, we always focus on the tumoral cells, and this was probably the big mistake of next-generation sequencing. Next-generation sequencing brought a lot of attention to the cancer cell, genetic and epigenetic alteration, whatever is happening in the tumor cell. But we did the mistake 10 years ago of thinking of tumors as just tumoral cells. Now, as we move forward with the role of immunotherapy, we look more about the connection, interaction between tumor and microenvironment. And here, I think it's important to underline the importance of genetics also and its impact on the surrounding microenvironment. And Sumera will talk thoroughly about this in the next talk. I just wanted to briefly introduce and show some unpublished data for our study. So recently, with this idea of connecting all the dots, or at least trying to do better than what we did before and focusing more on how the tumor connects with the microenvironment, we generated a new immunogenomics classification. I won't go into details to the 5 classes that are here indicated with different colors, but we interestingly observed that different genetic alterations were enriched in different subtypes, suggesting that, again, according to the genetic alteration, you will find a certain immune microenvironment rather than another. And interesting, going back to the FGFR2 fusion, as you can imagine, is one of my favorite targets. This was enriched in this class here indicated in green, which was kind of immunosuppressed. There was a lot of macrophages overall. There was a lot of immunosuppressive mechanisms. They were called tumors. And it was kind of interesting when we think forward on how to improve responses, would it make sense to combine FGFR inhibitors with immunotherapy and so on. But of course, we know very little, again, about this connection of genotype-immune phenotype. And trying to understand a little bit more, we looked at our cohorts comparing FGFR2 fusion positive patients versus FGFR2 fusion negative. And interesting, when we looked at the immune composition of the tumors, we observed important differences. In particular, we observed that those patients with FGFR2 fusion positive, they showed a lack of T helper cells, but they showed a significant enrichment of neutrophils. This was an immune deconvolution in vivo, in silico, which has its limitation. Therefore, what we tried to do was to replicate this into mouse models. And this has been done in my lab by 2 very talented master students. What they did is using the transposon system to try to model or generate tumors carrying FGFR2 fusions. And just for those of you who may ask, we tried both with human fusions and the murine homologue of FGFR2 fusions to avoid the mistake or the doubt of whatever was induced was because we were expressing something human into a mouse. And interesting, when we combined FGFR2 PPHL1, that is the fusion that we identified almost 10 years ago, when we combined it with activated YAP, we obtained intrahepatic cholangiocarcinoma within 4 to 5 weeks. As you can see here, the injection of the single gene, as we already know, is not sufficient to induce tumors. And interesting, more recently, we have performed flow cytometry analysis of these FGFR2 PPHL1 YAP tumors and compared to others that carry YAP but not the fusion or healthy livers. And if you can appreciate this synapse color in the FGFR2 fusion positive, those are neutrophils. So again, we observed that tumors expressing fusion have an abundance of neutrophils compared to healthy, but also compared to other cholangiocarcinoma carrying YAP, but in this case, not FGFR2 fusion. At the same time, also we observed a decrease in T helpers, but I have to say that given the abundance of neutrophils, we are giving priority to try to understand the subtype of neutrophils by single cell and other technologies, and try to understand how we can use this information to treat these patients better, and perhaps a combination with immunotherapy that might deplete or enrich these neutrophils depending on their function. Of course, genetics is important, but as we said before, there is a lot of challenges in accessing tissue. Therefore, I just want to mention the importance alternative of liquid biopsies. We know that now with the 2 blood samples, we can detect the most frequent genetic alteration. A lot of groups have been successful, and I think the liquid biopsy should be incorporated into management of patients, especially when consecutive biopsies are not possible. Of course, there are challenges. This is a study from Lipica Goyal highlighting the challenges when it comes to detecting FGFR2 fusions in the blood. And indeed, we still miss a bunch of those fusions using just blood, and the tissue is still indispensable. So in conclusion, of course, as I hope that I have conveyed the message, the next generation sequencing has been a great evolution and improved the management of these patients, but also has highlighted the huge heterogeneity that we have. We have identified distinct recurrent alteration, particularly for intrahepatic cholangiocarcinoma, that have had a great impact in terms of management. And of course, since the genomic profile is governed by underlying etiology, it's not surprising the presence of certain genetic alterations show variability across different geographic regions. What I want to highlight, and probably this is the best introduction for the next talk, I think there is a lot more that we do not know when it comes to genetics and interaction with the surrounding microenvironment. And I'm really looking forward to hear more from Sumera in the next one, and how we can use this information to improve treatment and combination strategies. I would like to thank my lab for doing all the work they have been doing. I would like to highlight Emily Brammel, who is a talented student who has a poster here, and her poster and her talk tomorrow will show how we're trying to go beyond genetic alteration and try to connect tumors with stroma, without, of course, taking the importance of genetic alteration, but we need to find more and do more. And of course, all of you for your attention. Thank you. And now it is my great pleasure. I already introduced a little bit, right? There was a little bit of spoiler alert about the next talk. It is my great pleasure to introduce my co-chair, Sumera Ilyas. We already heard about her accomplishments. I just want to focus that she has an NIH-funded lab, and she works particularly in immunotherapy and immunobiology of cholangiocarcinoma. And the title of the talk is Tumor Genotype-immune Phenotype Relationship in Cholangiocarcinoma.
Thanks very much, Daniela, and thanks for such a great talk. Let me orient myself. So we've seen this classification, anatomic classification, in several talks today, so I won't spend much time on that. So what do we know about the tumor genotype and immune microenvironment of cholangiocarcinoma? We know that actionable genetic alterations are present in about 40% of intrahepatic cholangiocarcinomas, or ICCAs, and approximately 15 to 20% of perihilar and distal cholangios. We know that immunotherapy has been incorporated into the first-line regimen for cholangiocarcinoma based on TOPAZ-1 and KEYNOTE-966 trials. And we also know that the response to immune checkpoint inhibition monotherapy has been disappointing. And there are several transcriptome-based immune classifications of cholangiocarcinoma. We heard about one of those from Daniela, and that was the classification that she had published. So what are the unmet needs? The unmet needs are that we haven't been able to correlate, I think to a great degree of detail, the tumor genotype and the immune microenvironment phenotype. And this is important because not only will this information inform development of new combination therapeutic strategies, but this can also help stratify patients for existing therapeutics. So there are, as I mentioned, there are several immune-based classifications that are of intrahepatic cholangiocarcinoma that are based on transcriptomic data. This is a classification from a French group that was published in Hepatology a few years ago, and they described 4 classes. And consistent with what we know about cholangiocarcinoma, that these are poorly immunogenic tumors. As you can see, where's the pointer? What did you use? Oh, right there, okay. It's on the wall. It's on the wall. As you can see, that the majority of patients were classified into the immune desert class, which had weak expression of immune and myofibroblast signatures. And then this is a classification that Dr. Sia had published. This is the stroma, tumor, and immune microenvironment, or STIM classification. And consistently, the majority of the patients were classified in classes that had poor, low immune infiltration. This is more recent data from the NCI based on the NCI Clarity study that integrated transcriptome and genome assessment of FFPE specimens before and after immunotherapy. And there were a total of 86 patients with hepatocellular carcinoma and cholangiocarcinoma. And they described 4 clusters, and each cluster was correlated with differences in survival. So 2 clusters, patients had good overall survival, and clusters C3 and 4 were associated with poor overall survival. One of the clusters with good overall survival had a greater immune infiltration, so that is not unexpected, whereas cluster 4 with poor overall survival had enrichment of tumor-associated macrophages. And responders had greater immune infiltration at baseline compared to non-responders, so this is something that we do know occurs across cancers, that greater immune infiltration does correlate with response to therapy, immunotherapy. And they also correlated genetic variations and conducted whole exome sequencing as well, and found that tumor mutation burden, or TMB, was not significantly different across the clusters. There were certain mutations that were enriched in clusters, so mutation rate of P53, IDH1, KRAS was higher in cluster C4. This is earlier work that was published in hepatology, demonstrating that loss of BAP1 is associated with increase in B7H4, which is an immunosuppressive molecule. This work was in integrated immunohistochemistry with next-generation sequencing from resected specimens of 96 intrahepatic cholangiocarcinoma patients. And preclinical data, so what I showed you so far, this is translational data based on human tissue. Preclinical data has demonstrated that mutant IDH1 promotes suppression of CD8 T cell activity. There's inactivation of TET2 DNA, so this is work from Nabil Berdisi that was published in Cancer Discovery a couple of years ago. And IDH inhibition stimulates CD8 T cell recruitment and interferon gamma expression. And so this begs the question, are tumor genetics important when considering immune checkpoint inhibition therapy in patients? Should we be stratifying? So based on these studies that I just showed you, what have we learned? And what are the unmet needs? So the immune classifications, I think that, of course, they're an important step in trying to correlate tumor genetics and immune infiltration, immune signatures, but they do require prospective validation. For instance, what we need are clinical trials that would use these immune classifications and then correlate response with immunotherapy. And one of the challenges with classifications based on human transcriptomic data, there is significant heterogeneity across patient cohorts. So it can be really challenging to look at specific tumor genetics and correlate that with a specific immune microenvironment phenotype. And so that is an unmet need. And you will see that across these data sets, there are, there may be the same genotype would be associated with a different phenotype, and I think that's due to the heterogeneity. And so what we need, this is a scenario where we think that, where I think we have to step back and go back to the bench and leverage preclinical models, particularly preclinical models with different genetic drivers, because that may permit a more stringent analysis of the tumor genotype and immune phenotype correlations. And so in order to do that, we need models with different genetic drivers. And a few years ago, we had published a biliary installation model. This is a cholangiocarcinoma model of carcinogenesis, employing biliary installation of YES-associated protein, or YAP, which is an oncogene, and permissive activated AKT, and coupled with the systemic interleukin 33. So this model was published in Hepatology, and more recently we've generated we generated a second model using a very similar approach, a Sleeping Beauty transposon transposase approach, where we instilled a dominant negative FBXW7. So FBXW7 is mutated in approximately, this is a tumor suppressor that is mutated in approximately 35% of cholangiocarcinomas. And so we instilled these and permissive AKT, and the reason I say permissive AKT, back in, when we had that, when we published the hepatology paper, we had shown that AKT alone, installation of AKT alone would not generate tumors. We did get some tumors with YAP alone, but AKT had, that permissive AKT had increased the tumor incidence rate in those mice. And so we, by doing this, we have tumors in about 16 weeks, and these are tumors that recapitulate key features of human cholangiocarcinoma, including malignant gland hyperplasia, desmoplasia, alpha smooth muscle actin expression, indicating the desmoplasia that cholangiocarcinomas are known for, keratin 19 expression, and lack of HepAR1, which is a marker of hepatocellular carcinoma. And then from these tumors, from these biliary installation model, the tumors that were generated, we were able to isolate a cell line, and that cell line also had, as expected, key features of human cholangiocarcinoma and the known markers such as keratin 19. And SOX9. And we were able to take the cell line and implant it in an orthotopic fashion into livers of mice, generating a syngeneic model. So from the AKT-YAP tumors, we had generated a model that we published a few years ago. It's termed the SB1 model. And so we generated this model in a similar fashion, and we termed this FBXW7 AKT in cholangiocarcinoma, or FAC. And then the model, as you can see here, has, so the cell lines as well as the syngeneic model has features of a cholangiocarcinoma, including malignant gland hyperplasia, desmoplasia, and alpha smooth muscle actin expression. And to compare these models that have different genetic drivers, so SB1 with YAP-AKT, FAC with FBXW7 dominant negative, and then we have a third model that we received the cell line from Nabil Berdisi. It's a KRAS P53 driven model, and we were able to implant that as well in an orthotopic fashion. So with these, to try to get a sense of their transcriptomic differences, we conducted bulk RNA sequencing and found and compared it to normal mouse cholangiocytes. And as you can see here, that these all three cell lines have transcriptomic differences. To put our findings into clinical context, we also conducted whole exome sequencing. And as you can see here, the KPPC cells have a very high tumor mutation burden of 50.83, and we compared the whole exome sequencing data of the three cells with the known human cohorts. And FAC and SB had a similar TMB to what we see in different human cohorts, whereas KPPC was significantly higher. And then we assessed known mutations in human cholangiocarcinoma and found that these were present in the mouse models as well. So this is the human data, and this is the mouse data. Next, we took these models, took these cell lines and implanted them into mouse livers. And the tumor rate does vary. The KPPC model is more aggressive with much higher tumor burden compared to the SB1 or FAC. And then we conducted single-cell RNA sequencing as well as site-seq and confirmed those results with the flow cytometry. And what we learned from this data is that these models that have different genetic drivers, they do have diverse immune microenvironments. So for instance, the KPPC model, there's a preponderance, it's a very monocytic model. There's a preponderance of tumor-associated macrophages. They also have a lot of MMDSCs compared to the SB1 and FAC tumors. And on the other hand, the SB1 model is more granulocytic, and there's a preponderance of granulocytic or PMN MDSCs in this model compared to KPPC. The FAC model has more of a T-cell infiltrated immune microenvironment compared to the other two models. And there were a higher abundance of CTLs as well as effector T-cells with the granzyme B production in the FAC tumors. So why is that important? These varying immune microenvironments, what does that mean? So to dissect that further and see and place that in a therapeutic context, we treated mice, tumor-bearing mice, that had FAC cells implanted, KPPC or SB1, and we implanted them in mice where the extracellular domains of PD1 and PDL1 have been replaced by human PD1 and PDL1, and that allows us to treat these mice with FDA-approved immunotherapies, such immune checkpoint inhibitors such as pembrolizumab, nivolumab, and durvalumab. And when we did that, we found that, and of course we've done the same set of experiments, and this was reported in this paper that was just published in Journal of Hepatology. We did the same set of experiments using mouse anti-PD1 and PDL1 as well, and had fairly consistent results. And so we found that the FAC model, FAC tumors which have more of a T-cell infiltrated immune microenvironment, they responded to immune checkpoint inhibition with nivolumab alone. So this is monotherapy. Similar findings with KPPC tumors, they responded to ICI monotherapy. So these are tumors, again, that had a very high TMB. And so I think that's the reason that they had a response to ICI monotherapy. And then the SB1 is our poorly immunogenic model. This is a model that had a preponderance of immunosuppressive myeloid cells. And as you can see, and we've previously shown, this was work that we published a few years ago in JCI, that this model has a preponderance of immunosuppressive elements and does not respond to ICI monotherapy. And we confirmed that here in this paper using durvalumab, that these tumors did not respond to ICI monotherapy. And so in summary, we have, with this work, I think, we've taken a step towards trying to define the tumor genotype and immune microenvironment phenotype relationships using models that have different genetic drivers, correlating them to human CCA, and interrogating their immune microenvironment and response to immunotherapy. So is tumor genotype the end-all, be-all for dictating the immune microenvironment phenotype? No, because that would be an oversimplification, and there's nothing in cancer that is simple. There are phenotypes that are more germane to cancer biology that still matter. So while we think tumor genotype is important in influencing the immune microenvironment, it's not the only factor. And so this is the reason I showed this. It's an example of why that is so. So this is work that we also recently published earlier this year, and in here we demonstrated that non-canonical tumor necrosis factor-related apoptosis-inducing ligand, or TRAIL, promotes tumor growth in cholangiocarcinoma. And so the TRAIL-R system has been extensively studied in cancer biology, but more so as a pro-apoptotic factor. But all TRAIL agonists have universally failed in human clinical trials, and we think this is part of the reason, because the role of TRAIL signaling is very complex in cancer biology. It's also very context dependent. And in cholangiocarcinoma, there is it's very clear to us now that TRAIL signaling has a dual role, but a very potent immunosuppressive role, leading to an increase in myeloid-derived suppressor cells and CTL inhibition. And so the tumor genotype did not matter here, because when we took mice that tumor-bearing mice that were SB tumor-bearing, KPPC, or FAC, and implanted those cells into wild-type mice or mice with a TRAIL-R, total body knockout for TRAIL-R. Regardless of the tumor genotype, there was a significant reduction in the tumor burden across all the models, and this was mainly due to an abundance of MDSCs, and so this, I think, highlights that tumor genotype is important, but it's not everything, and this work demonstrated to us that non-canonical trail signaling promotes tumor immunosuppression in cholangiocarcinoma. And so key take-home points are that it's, as Dr. Sia also alluded to earlier, it's really important to understand the tumor genotype and immune microenvironment phenotype. It's an unmet need, and it's something that clearly has therapeutic implications. Existing human transcriptomic data has taught us that genetic alterations such as IDH1 mutations, BAP1 loss, these are associated with immunoinvasion and tumor suppression. And preclinical models are one way to try to really dissect these correlations between tumor genotypes and the immune microenvironment phenotype. And our work has demonstrated that mirroring models that have distinct genetic drivers, they do have diverse immune phenotypes, and that does correlate with the response to immune checkpoint inhibition. And this data also suggests that ICI is not a one-size-fits-all approach, and we should consider stratification of patients according to their genotype. So with that, I'll conclude. That's our team, and Rory showed our CCA group earlier today. So there are 6 PIs, and 4 of us are here at the Cholangiocarcinoma Foundation. So thank you very much for your time, and I look forward to the discussion.
Given the prevalence of HRD and really the question about whether those mutations are functional or driver or not, I know many patients ask, should they have something like the Myriad diagnostics or a test like that would be maybe a predictor of HRD, just to see if something like a PARP inhibitor would be beneficial. I know there's an absence of data, but for patients, I think they don't really have time to wait for the trials to read out. Do you recommend that, or how are those tests? Are they different for cholangio than ovarian, or are they predictive?
So I definitely recommend testing germline for an HRD mutation if the tumor holds a candidate germline mutation that could, say, a tumor BRCA1. Definitely that patient should be tested for germline with a validated panel. The HRD signatures in tumors have not yet been validated specific to biliary tract. They've been really focusing on, obviously, the more prevalent tumors, breast, ovarian, et cetera. But I do know companies like Tempus are working on expanding their HRD signature validation in other tumor types. So I think right now the things that would prompt me to refer, which do prompt me to refer a patient for germline testing, are finding a candidate hit in the tumor, and the point that I said earlier, which would be a young age, personal or family history of a different cancer, and I think those are I'm missing one of my criteria, but yeah, age under 50, tumor mutation, and personal or family history.
Yeah, great talks, everybody. I guess my question is for Sumera, and the really cool mouse models you have where you've replaced the human extracellular PD-1, those are going to be incredibly valuable, first of all, so just a comment. And, the question I had was related to the different stromal and fibroblast architecture across the models. Have you guys looked at that? And do you think that that in any way influences the differences in immune profile that you see between the 3 mouse models?
Yeah, that's a great question. And that's something that we are very interested in. So for the work that I presented, we really focused on the CD45 positive populations. And now we're working on conducting single nuclear RNA sequencing, because I think that will allow us to look at that crosstalk between, I do think it is very important. I think CAFs are very important. Some of the work that we've done earlier already shows that there's significant crosstalk between cancer-associated fibroblasts and immunosuppressive myeloid cells and cholangiocarcinoma. So I think these will be crucial. But this was a first step towards trying to get a better understanding and correlating the genotypes and phenotypes. But that doesn't mean we stop with the immune microenvironment. I think the stromal components are very important.
Tim has a question.
Thank you, Katie. Every single time you give a talk, I learn something new, so it's amazing. But what are we going to do if we actually identify these mutations in a patient? So, in other words, so let's say you have a positive individual, which is not a patient yet. Obviously, I used the wrong word. We don't have any idea how to screen for these patients, how to do surveillance for these patients. And how is this going to work?
So you mean if we find a germline
yes,
BRCA1
let's say,
or something like that?
Right.
Well, I would say you're right. We don't have a way to yet screen for cholangiocarcinoma, but we do have preventive and screening for breast, ovarian, et cetera, and those are the ones that are super high penetrant. So we don't want to miss out on diagnosing a patient or a curable patient or a family member with something like a BRCA1 or BRCA2 or because there are other tumors for which we do have tractable screening and prevention mechanisms.
So you would dismiss the risk for cholangiocarcinoma?
I don't dismiss it. I would say that it is an evolving work in progress. And I did mention that I think that would be a topic we should invite some interventional endoscopists to present their EUS data for, obviously pancreas is in the same boat and that's a more difficult, well, actually the same boat where you certainly, if you can prevent a breast cancer, let's do that. And in parallel, work on our early detection of biliary and pancreas and the more difficult ones.
If I could add a little granularity to that question. So you have a patient who has a germline variant. It's important to screen them and their family members for other tumor types. But to put into context how you would think about screening, what is the lifetime risk as you've reviewed the data for someone with a pathogenic germline mutation, BRCA, in breast versus pancreas versus cholangiocarcinoma?
So I think for BRCA and breast, it's like 60%,
Right.
Ovarian 40-ish, depending on which one. And then the breast cancer linkage analyses were suggesting maybe a 5-fold increased risk with really small numbers of biliary tract cancer, so certainly not as high, being a much rarer tumor type overall.
So that's 5-fold over population.
Over the general population.
So
small.
from sub 1% to,
And
so
then-
still a relatively small, back to Tim's point, it's still probably a relatively small lifetime risk.
Correct.
So we have to think about it with the advanced endoscopists, as you say.
But
how you
the
do
motive for screening is to get the high yield, prevent breast, prevent ovarian.
that. Right.
I actually have a question while you're bringing the phone. I don't want the germline conversation to take over, but I guess piggyback on that to Dr. Kelley. I wonder if, because this is all new to me, I work in ovarian and breast cancer quite often, and we actually look at pre-malignant lesions. And I was wondering if there's any evidence of any of these lesions in colorectal carcinoma, like STIC or DCIS in ovarian and breast.
Well, there are certainly intraductal neoplasms in the biliary tract, and I don't know their relationship to germline mutation status.
It's not known. It hasn't really been studied. Yeah.
They can have varying degrees of dysplasia, but I don't think they've been consistently genotyped. To answer that question, it's a great question.
Thank you again for your presentations. I think we're increasingly accepting the concept that the environment that the cancer is in is a dynamic environment. So immune cells will come and go, and the percentages will change. Epigenetic environment will be different. So how confident are we about making a decision on a single time biopsy on a patient? And how do you see that changing going forward in the clinic? I know it's a difficult question, but I think it's something we need to address. This is to anyone on the panel.
I can start. I think it's a crucial question, right? Because so much of the scientific world has focused on snapshots, right? And even those snapshots are so complicated because there's so many different components and context. I mean, we've learned about all of this today, right? Context matters, tissue specificity matters, the genotype-phenotype relationships, immune phenotype relationships matter. And so, similar to what I was saying, how I responded to Dr. Lazinski, I think looking at the snapshot is the very first step. I think we'll really start to make meaningful progress when we're looking across time, when we're looking, particularly after therapeutics, because that also is a key factor. You know, all of these factors are modulated by therapies as well. So I think that's critical. And then the differences between primary and metastases. I mean, it's just there are a lot of factors. I think they're all exceedingly important, which is also why I truly believe it will be the collective efforts of all of us that will ultimately make a difference.
Maybe I can add something. We say, these are snapshots, they're not enough. And still we don't have enough snapshots, right? When it comes to the new immunotherapy in the standard of care, and we need desperately predictors of response or resistance to try to better select patients. Still, we do not have enough pre-treatment tissue biopsies to kind of start digging into which could be, the right type of patients to receive that. So yeah, there are snapshots that are insufficient, but they're crucial. And we see a lot of patients with cholangiocarcinoma, still we have a lot of challenges get biopsy pre-treatment. So I mentioned in the talk liquid biopsy, that might be an answer. It doesn't look at the whole features that we need, something to take into consideration, and I hope that we will, as we move forward, we will have better ways of getting this information on treatment from each single patient.
Hello. My name is Sadapan, one of the Hemon Fellow from Boston. I have a question for Dr. Ilyas about TMB. So in your mouse genetic knockout models, what do you think is the driver of a higher TMB in that particular model, when in humans, really the driver of higher TMB is more about carcinogenic exposure, like radiation or smoking? But in the knockout models, like what would be the driver of those differences?
You mean in the KPPC model?
Correct.
Yeah, I think that's a great question. It's not something that we really delved into in that paper. And that was the only model that had a really high TMB. My personal bias is anytime you mess with P53, you will create chaos. And so in part, I think it's a reflection of that, why that model had a really high TMB. But did we specifically look into why so with those two drivers? No, we did not.
Hi, I had a question for Dr. Maia. I thought it was so interesting you were describing these macrobound enhancers in ovarian cancer, and just wondering, have you looked in other cancer types? And you showed that there was regulation of the estrogen responsive gene expression program. What might those enhancers regulate in other tissue types, such as cholangiocarcinoma? It might be speculative, and also because I'm interested in chromatin remodeling, do you know anything about how chromatin remodeling complexes interact with these macrobond enhancers?
So the macrobond analysis that we did initially was actually in breast cancer. That's the initial analysis. But actually, my lab currently is looking in ovarian cancer, and we're very interested also because one part that I didn't get into is that the macro domain is actually very similar to the PARP, the macro domains that PARP have, PARP proteins. And so we actually believe that macro H2A could have some sort of relationship with PARP response, because it is also an inhibitor of PARP1. So we're looking into that in the context of ovarian cancer. In terms of chromatin remodeling, actually, the reason why I got into histone variants is because I was really interested in chromatin remodeling. But there's really it's not very clear. There's 3 different isoforms, and so we still really don't know exactly how they're affecting. And it's really complicated because macro H2A was actually initially identified in X chromatin, so in the repressive chromatin of within axon activation chromosome. But this effect that we see in enhancers could be masked sometimes by repressive effects in the gene bodies itself. So it's really kind of complicated. We've been designing some CRISPR-Cas tools to really bring macro to specific enhancers and try to define whether those effects are enhancer specific or not. But it's still kind of a field of research that we're looking into. I would love to collaborate and look into cholangiocarcinoma for sure.
Can I ask 2 questions? I have one for Katie. You showed the beautiful subgroup analysis in the TOPAZ-1 trial, where there were all the genes. Was that based on whole exome sequencing?
No, that was based on, I believe, a foundation panel.
Okay. Do you have enough depth? I would say enough, information about other genes to look into different. Like, for example, I was interested in KRAS. There seems to be not an association, right? What about KRAS, depending on which other alteration it occurs, right?
Right, co-mutations and such.
Yes.
I don't know that, so I think about roughly 400 of 600 or so, 2/3 or so of the patients on TOPAZ-1 were in the biomarker cohort that had a clinical panel. I think it was Foundation 1, next-gen sequencing. Their sample size for any individual hit is going to be very small, but the clear area to go is to ask, not that this is easy to do, but there's the Keynote 966 database that was over 1,000 patients that also has paraffin and would, more than double the sample size. And the pooled analysis could start to interrogate at least co-mutation data with this kind of profiling that's been done. And there are bank samples too.
Yeah, great talks and great data. I have a question to Daniela. This link of FGR fusions with neutrophil infiltration is really interesting. Have you looked deeper into that? What are the signals that recruit the neutrophils? Are there differences in the secretome of these the specific genetic subtypes, so to say?
So we're just starting. So we're able to generate the mouse model. As I mentioned, one of the first concerns was to express a human FGFR2 fusion. So we wanted to make sure whatever we were observing in terms of immune phenotype was not due to the fact that we were expressing a human gene. So it took a while to generate the immune homolog, but we see exactly the same. There is this enrichment in neutrophils. We're trying to apply now other technologies to kind of dig into the subtype of neutrophils, what type of neutrophils they are. We're trying single cell, but I know there are some challenges in detecting neutrophils with single cells, so we're also designing an extended flow cytometry panel to dig into the specific subtypes. And as I said, I'm very interested. It seems to be something associated specifically to patients overexpressing FGFR2 fusions. We confirmed this in the mouse. When it comes to the patients, we had to put together a lot of patients because, as you know, the frequency, it's 10-15%, so you never know if the association is strong enough, and we don't have enough data to replicate it in other cohorts, so we have only one. The fact that we replicate into the mouse, I believe there's this valid association. How the mechanism and why it's happening, I do not know, but it gives me hope that these neutrophils have an immunosuppressive role, and they are facilitating, by depleting them, we will actually improve responses to FGFR inhibitors, other immunotherapies.
Thanks.
I have another question about pre-malignancy. I know we're talking about cancer, but in the pancreas world, it's, I think, come to everyone's attention that there's a very high prevalence of pre-malignant lesions, Pannin lesions, KRAS mutations in the general population, people who won't go on to develop pancreas cancer, but at least now we know that a lot of people have them. As Katie pointed out, we don't really know enough about the precursors to cholangiocarcinoma, but we do a lot of liver biopsies, especially in people who have cirrhosis who are at risk. So should we be looking at our liver biopsies for these mutations, for epigenetic changes, or is that a needle in a haystack kind of thing to think about doing?
So I don't know much about the liver, but I do think that it's very useful. We've been doing a lot of single cell sequencing in early lesions in ovarian cancer, and it's actually, if you look at germline patients, even before we know that they had lesions, we actually are able to identify several of them. So I think it's very useful. One thing that we actually are very interested in looking at is the microenvironment, how different the microenvironment in these lesions is between germline mutants and not. And we actually see some differences. So it is possible that actually this predisposition also would affect their ability to respond to immunotherapy, which I think will be a really interesting field.
I think I think in the context of the liver, it actually would be a needle in a haystack type of situation. So the premalignant lesions in cholangiocarcinoma, this is bilin, and which Dr. Sauer talked about. A lot of the GEMs have that, although we don't really see them in our carcinogenesis models. But when the way we pick these lesions up in clinical practice, it's usually more endoscopic, findings that would lead to that, not really a liver biopsy. And they're overall very rare. And I think that spectrum is just not very well defined in terms of, I think it's hard to say that majority of these would have, unlike pancreas, that majority of these would originate with, let's say bilin and go on to that. And so that's why I think if we just did, let's say, liver biopsies, it'd be harder. Do you want to add to that comment, given your expertise?
Sorry, hi. From a perspective of surgical pathologist, I have to say, the premalignant lesions, the problem is they're mostly extrahepatic, and the extrahepatic are hard to get, but they're not rare at all. If you look at resection specimens, you have 80%, more than 80% bilin, high-grade bilin in the extrahepatic field. Intrahepatically, with the small duct, you are aware that there aren't any precursor lesions, so you won't see them because they are not defined yet.
Right, and that's why liver biopsy wouldn't pick them up, but it would have to be endoscopic approaches, because when we do see it, it would be more intraductal, and it would be for the extrahepatic types.
Exactly. So if you want to do research on them, you only can do this on a resection specimens.
All right, I think one more question.
My name is Masafumi Ikeda, National Cancer Center Hospital East Japan. So I have one question for Katie Kelley. So we are now initiated a phase 2 trial of the PARP inhibitor in second line, later line setting for the BRCA mutation. However, my impression is not so good. So because of the first line treatment is gem platinum already done. So and almost all patients progress the gem platinum. So I think the PARP inhibitor is maybe my idea is that so maintenance treatment strategy is better like the pancreatic cancer. So do you have any idea to how to use the best strategy of the PARP inhibitor?
So were you suggesting it should be used earlier before platinum resistance?
Yeah, right.
Yeah, I think that's a great point, that in our first line therapy, well, a couple points, I guess. One is that there is a place out for first line in the maintenance setting where patients may not have progressed on a platinum and would be, akin to the polo context, be a really intriguing population to look at PARP inhibition. But second point is our use of platinum in biliary is a very low dose, and we restrict it to just a window of 6 months. So we don't have a definition for platinum resistance in terms of like the conventional ovarian definitions. And that's something I think is an area that is necessary to move forward with PARPs. And then the third point is I showed the Hokkaido cohort, I believe, from Japan, that only about a third of BRCA patients had a second hit or were true HRD. And so this is where our trials are going to potentially miss a signal if we're diluting, if let's say we enroll 40 patients with BRCA type, either tumor or germline, but we don't know which ones have true both, we're going to miss a signal. They're going to look negative.
Yeah, half of the patient is a somatic BRCA mutation, and half of the patient is germline mutation. Biliary tract cancer is slightly different for the pancreatic cancer.
Okay, I want to thank the speakers for a really great session. Thanks everyone.
1 second, just a couple of housekeeping. So we're breaking for lunch right now. We will reconvene here at 1. Team Greten will deliver the state of the art. Just a reminder that we have the Young Investigator Dinner tonight from 6 to 7:30 in room 250A. See you here at 1. Thank you.